Very good afternoon. I'm Aisha and I will be your host for today's webinar. Kindly note that this webinar will be recorded. It is my pleasure to welcome you to our monthly ACTRIS Cell Therapy Lecture Series. Today's lecture focuses on progress and challenges in retinal cell therapy for vision regeneration. Let's welcome Assistant Prof. Su Sin-Yi, our distinguished speaker for today's webinar. Prof. Su is an ophthalmologist and vitreoretinal surgeon at the National University Hospital of Singapore. She is the Divisional Director at the Institute of Molecular and Cell Biology and Research Director of the Department of Ophthalmology. She runs a broad research program in retinal diseases as Senior Principal Investigator of Translational Retinal Therapeutics Lab at INCB and clinician scientist at the Singapore Eye Research Institute. Her research in retinal therapeutics has been published in Nature Biomedical Engineering, Advanced Materials, Nature Communications, amongst others. She received Asia Pacific Vitreoretinal Society LDP Gold Medal Award in 2021, Eye and Vision Health Distinguished Award in 2021, and Asia Pacific Academy of Ophthalmology SSO Young Ophthalmologist Award for 2 consecutive years, 2019 and 2020. Prof. Su currently leads several translational research programs on retinal cell therapy. Just a little housekeeping before we get started. If you have any questions during the presentation, please type them into the Q&A window. Prof. Su will address these questions after her presentation. And now, without further ado, I will hand the time over to Prof. Su. Prof. Su, please.
Thank you, Aisha, for the wonderful and kind introduction. And thank you, ACTRIS, also for the kind invitation to speak at this forum. It is indeed my pleasure and my honor to share with you some of the work that we have done over the past few years, as well as to give you an overview of the current landscape for retinal cell therapy. So just to give you an overview of the talk, I'll be giving you a little bit of an introduction of what the retina is for the non-ophthalmologist amongst us. And then I'll be dividing my talk into 2 parts, the first of which is to look at retinal pigment epithelial cell transplant for the treatment of age-related macular degeneration and some of the work that has been ongoing in my lab. And the second part, we will be focusing on retinal progenitor cell transplants for the treatment of retinitis pigmentosa, which is another condition, and some of the work that we have done in our lab. So as a retina specialist, I find that the retina is a very fascinating part of the eye. And this is because perception of vision begins first at the retina, and that continues the visual cortex of the brain at the occipital lobe. And the retina is fascinating because it is indeed the window to the brain and to the eye. And light is converted to electrochemical signals by phototransduction in the retina. And I often describe the retina as the gui lapis known to my patients because in the retina, you have multiple layers of cells and it's a really complex tissue. It's broadly kind of categorized into 3 areas or 3 main layers. The most important one, of course, is the photoreceptor layer, which is shown over here. And it's important because it receives and transduces light energy to action potentials. And then these action potentials are then transmitted to the bipolar cells, which is the middle cells over here in blue, before being transmitted into the ganglion cells, which then project to the thalamus and then the brainstem for vision. And there are also 2 other tangential sets of cells, the horizontal cells over here, as well as the amacrine cells over here. And in this talk, we're going to focus primarily on photoreceptors, which are the cells over here, as well as the retinal pigment epithelial cells, which is really important also to support the healthy function of the photoreceptors. So this cartoon over here shows the retinal pigment epithelial cells over here. They are cuboidal and they are polarized with the apical region as well as the basal region. And the apical region is abutting the photoreceptor. This is because it plays a very important role in photoreceptor renewal. It phagocytoses the outer segments of the photoreceptors and recycles that, and this ensures photoreceptor health. It also secretes goodies such as PEDF as a neurotrophic factor to support the function of the photoreceptors. And the basal layer, it secretes VEGF to support the vascular function of the choroidal plexus. Importantly, it also forms tight outer blood retinal barriers over here, and this prevents inappropriate accumulation of fluid within the retina. It sits on a Brooks membrane, which is a basement membrane layer. It also plays an important role in retinoid processing and recycling. It is highly pigmented because it has melanin granules, and this is important because it allows absorption of stray lights. And the first disease that I really want to focus on is age-related macular degeneration, also known as AMD. AMD is a leading cause of irreversible blindness. It is the third cause of blindness globally, and it afflicts particularly the elderly population. And it is estimated that 8.7% of the population between the age of 45 to 85 will have AMD of various degrees, whether early or late. And there will be an increasing prevalence of late AMD as one ages. And this is reaching a level of global epidemic because of our global aging population, as we are all familiar with. Now there is an estimated incidence of AMD of 288 million by 2040, particularly of a high incidence in Asia. This is what patients would experience if they have age-related macular degeneration. They'll start to lose their central vision, as you can see over here, and over time that would progress. Soon they would be unable to see the faces of their loved ones or be able to do daily activities such as reading or writing. So broadly speaking, age-related macular degeneration can be divided into three main groups. So you have your dry and the wet. And the wet is so-called wet because there is bleeding involved due to development of abnormal vessels in the subretinal space. And this is called choroidal neovascularization. And there's a particular subtype in Asian populations, particularly called the polypoidal choroidal vasculopathy, where there is a massive submacular bleed. And this can be seen or followed up in clinic when we do a optical coherence tomography scan, as shown here on the left, instead of a normal retina with a normal foveal contour. But on the right is a case of what is quite typical of age-related macular degeneration, where you have accumulation of subretinal fluids and hyperreflective materials, as well as irregularity in the retinal pigment epithelial layer. Fortunately, for the wet type of AMD, there is currently treatment. In the clinic, we do give frequent intravitreal injections of anti-VEGFs of various sorts into the eye, and this has been shown to reduce the onset of blindness due to wet AMD. However, there is still a subset of patients that eventually do not respond to treatment and still progress to the formation of disciform scar, as you can see over here for wet AMD. And particularly for dry AMD, there is no treatment yet because, as you can imagine, anti-VEGF would not be helpful for this form of AMD where neovascularization is not part of the disease pathogenesis. So there is still an unmet clinical need for patients with end-stage age-related macular degeneration. And so because retinal pigment epithelial cells, as I've highlighted to you earlier, play such an important role in the support of the photoreceptor function, they've also been found to be important in the disease pathogenesis of age-related macular degeneration. And RPE cell dysfunction and cell death is actually a hallmark of AMD. And thus, it is an intuitive approach for us to develop stem cell-based RPE therapy. And the concept is pretty straightforward in that you've got a stem cell, either embryonic source or induced pluripotent cell source. You culture them in the lab and differentiate them into retinal pigment epithelial cells, allow them to mature over time, in which they will start to develop pigmentation. And once you've characterized them, you can transplant them into the eye by various methods. This is an overview of some of the more high-profile clinical studies for RPE cell transplants. By and large, they have shown to be safe and have certain amount of ability to improve vision. The first two studies highlighted here are using human embryonic stem cells. And the third one over here is using skin fibroblasts derived induced pluripotent stem cells. The first study done in London, supported by Pfizer, was done in 2015 and published in Nature Biotechnology. Here they are using human embryonic derived RPE cells grown on a polyester membrane. So here they are transplanting it as a monolayer rather than a cell suspension with a certain amount of local immunosuppression. They demonstrated that in 2 patients, there was 29 or 21 letters of improvement over 12 months. Unfortunately, in 1 patient, developed proliferative vitreoretinopathy, which is quite a well-recognized complication of retinal detachment surgery and also in cell transplant surgery, and that was repaired surgically and the patient's vision was maintained. The second project was a Californian project in 2018. Here, similarly, they used a human embryonic stem cells grown on paralin, and they gave oral tacrolimus immunosuppression. They performed this in 4 patients, of which 1 patient had 17 letters improvement, and 3 patients were able to demonstrate improvement in fixation. This was published in Science Translational Medicine. And the third study highlighted here is the study by Masayo Takahashi's group in Japan, whereby they used autologous skin fibroblasts-derived induced pluripotent stem cells going into a monolayer, but here they did not use any scaffolds, so surgically it was a lot more tricky. And they had 2 patients followed up for 4 years with no documented improvement in vision, but no adverse side effects. But unfortunately, the clinical trial was halted due to a mutation identified in one of the patient's derived cells. So as you can see, there are some limitations to current cell resources. For human embryonic stem cells, it's a limited cell resource. There are some ethical concerns over the fact that it's derived from embryos. And because there is, you can't actually HLA match cells, so the issue of immunosuppression is always there. For induced pluripotent stem cells, there is an issue of whether these cells are also safe because of the potential accumulation of mutations, particularly if you remove skin from aged patients to develop autologous cell resources, not to mention the high cost that is involved if you were to develop a specific cell resource for each patient. There is also the approach of using an allogenic induced pluripotent stem cells by using a cell bank. Of course, there is an ability for HLA matching and maybe perhaps the need for the less requirement for immunosuppression. It is also not clear out there in the field what is the best approach for RP cell transplant. Should we do a cell suspension as such, where you just have cells that are loose, they're not attached as an adherent or a monolayer, or would it be better to transplant them as a cell sheet? The pros of doing it as a cell suspension is that it's a much easier surgical procedure, but as a cell sheet, it's more challenging to deliver the cells. But in this case, the cells are mature, they are polarized, they are functional, and therefore there's the argument that it's easier for them to function well in the in vivo situation post-transplant, and therefore have a faster recovery as well as a better vision outcome. So as you can see, there are still multiple challenges remaining for RP cell transplant. As mentioned earlier, should we do a monolayer approach with better outcome, but surgically more challenging, and therefore outcomes may be curtailed by the fact that sometimes we have surgical complications? Or should we use cell suspension, where it's essentially easier, but sometimes as the cells reflux out of the injection site, you might have a higher chance of developing post-op complications like proliferative vitreoretinopathy. There is also a discussion of whether there is an ideal cell resource that is safe with low risk, whether there's cell resources that are more ethically ideal or not as limited as embryonic derived stem cells. And of course, the issue of cost effectiveness using an allogenic versus an autologous cell resource. And importantly, I think, although many of the clinical studies have shown a limited amount of efficacy, much of this is still reduced by the uncertainty in whether we need immunosuppression, because a lot of these rejections are still being observed. And if we were to use immunosuppression, would we use a systemic immunosuppression or a local immunosuppression? Is HLA matching sufficient to overcome some of this immune response? And we don't actually know in these clinical trials whether the cells are able to survive and integrate, because we don't have ex vivo or histology evidence to tell us. And also, at the end of the day, age-related macular degeneration is a complex disease involving RPE, photoreceptors, and Bruch's membrane. And one would argue that if you just solely transplant RPE, would you be able to have a sufficient efficacy of functional recovery? And so what our group has sought to do over the 5 years is to develop alternative stem cell resources that addresses some of these issues. So the first project is one in which we have collaborated with Professor Tim Blankenship. Here we have worked with him to develop an adult human-derived RPE cells. So these are cells derived from the globes of cadavers. And these cell resources are potentially ideal because they are more mature. They can be HLA matched, and because they are more mature, there is a less propensity for them to maybe de-differentiate in vivo or to develop epiretinal membranes, as I will show you later on. And the second project here is looking at developing a cord lining derived induced pluripotent stem cells that have some hypoimmunogenic properties, which again, I will share with you later. And the third project here is working with a biotech company called Retinal, which we are funded by ICP funding to form a joint lab to look at a blood-derived iPSCs cell bank that has already gotten FDA approval and therefore has been certified safe. And they are vastly characterized by karyotyping RNA sequencing to show that they do not contain mutations. And together with Retinal, we have differentiated their iPSCs into retinal progenitor cells and done proof of concept studies in non-human primates, which again, I will share with you in a later part of my talk. Now, the focus on the first project in which we have derived adult human RP cells, this has been recently published in Stem Cell Report. So essentially, Tim in New York harvests the cells from human cadaveric globes. He isolates them and grows them in vitro, matures them for about a month, and then he flies them over to Singapore where we mature them for about another month. And then we transplant them into non-human primate models and keep them in the non-human primate eyes for 3 months with systemic immunosuppression. And we do a monthly follow-up of OCT, FFA, AF, as well as enucleate the eye at the end of the experiment to look at the functions of the RPE in vivo. So the first thing we sought to do was to see if this human adult RPE monolayer can be safely transplanted into the submacular space of the non-human primate models. So this is something we did in collaboration with Boris Stencil, Gavin, as well as Professor Gopalingam. Here is a video showing you the approach for a cell suspension transplant. So this is typically what we would do, in which we would take a small bore needle and cannula and inject the cells into the subretinal space. But as you can imagine here, often we do see a reflux of these RPE cells out of the injection site. And some of these cells may accumulate on the surface of the retina and they would cause the epiretinal membrane as such, causing a distorted macula. So what we've done is do an alternative approach where we grow these RPE cells as a monolayer as such on a PET scaffold, and we've developed specific instrumentations in order for us to develop to transplant this RPE into the submacular space. This is a video of what we have done in a non-human primate model. Here we are using a cannula to create a subretinal bleb. This creates a space into which we can transplant the retinal pigment epithelial cells into the subretinal layer. Then we use the intraocular scissors as such to create an opening. Because as you can imagine here, we can't just inject the cells in with the needle. We actually need to create an opening in order for us to transplant the entire cell sheet into the subretinal space. This is the RPE scraper. So what we intend to do is to scrape the underlying healthy RPE away and onto that area of bare RPE, we would be putting our transplant on top. So this is our RPE shooter, which has been developed and patented. And here we are injecting the RPE implant on a PET scaffold into the subretinal space just under the fovea. And we are able to assess or confirm the position of the implant by doing intraoperative OCT. As you can see here, that the implant is sitting quite nicely under the retina with the retina lying nice and flat over it. So now that we've shown that we're able to safely transplant the monolayer RPE into the submacular space, we next ask the question if this xenograft is able to survive and mature in vivo. So this is a fundus picture showing the AHRP grafts in the eyes of the monkeys at 3 months, right just nice under the fovea. You can see that that is the scaffold on OCT with a layer of hyperreflective cells overlying the scaffold, probably indicating that our cells are alive. And then in the control where we transplanted scaffolds without cells, you can see that the overlying retina is beginning to become thinner, showing signs of atrophy. And we confirmed this by a histology where we enucleated the eye at the end of the experiment, and we showed that there was the presence of the adult human RPE monolayer over here, sitting on the PET scaffold with a nice thick layer of the retina with the multiple layers, as I've explained to you earlier. This is in contrast to the control where it's scaffold without cells. You see that the overlying retina is much thinner and atrophic, and much of the outer nuclear layer over here has diminished. And we quantified this on OCT. Here you can see that in the 1 month, the ONL thickness in the area where we've transplanted the adult human RPE is much thicker than that of the scaffold alone at 1 month, and this was maintained at 3 months. So this is the OCT images that we can acquire as a video, and we can quantify the volume of the outer nuclear layer as such. And this is a 3D reconstruction of the outer nuclear layer, both at 1 month and 3 months, showing that it is well maintained overlying the area of the transplanted RPEs. Next, we wanted to do an ex vivo analysis of the adult human RPE monolayer. So what we did here was we enucleated the eye from the monkey to do histology as well as immunohistochemistry, and we compared that to the pre-transplanted cells embedded for IHC as well. So you can see that in the pre-transplanted cells compared to the post-transplanted cells, in the post-transplanted cells, there was a higher expression of RPE-specific markers such as RPE65, as well as CREL-BP1, as well as OTX2, which indicates to us that these cells have continued to mature in vivo. And we show that it co-localizes really well with human markers such as TRA185, indicating that these are human cells that were transplanted and these are not the endogenous monkey cells. Next, we also wanted to see if our transplanted cells is able to support phagocytosis in vivo, which I mentioned earlier is a really important function of retinal pigment epithelial cells. And so we did this by labeling the cells with rhodopsin, the photoreceptor outer segments with rhodopsin, and the apical portion of retinal pigment epithelial cells with ezrine. And what we were hoping to see is if the rhodopsin is being taken up into the retinal pigment epithelial cells, you would be able to see green globules beyond the pink line in the cytoplasm of the retinal pigment epithelial cells. And indeed, this is what we saw. So as you can see here, green is the rhodopsin, labeled photoreceptor outer segments. You can see that some of these phagosomes within the area of where we would expect the RPE cells to be labeled by DAPI over here and the ezrine layer in pink over here, indicating that functional phagocytosis has happened in vivo. Lastly, we wanted to see if these mature adult human RPE cells indeed have a reduced propensity to undergo epithelial mesenchymal transition, EMT, and therefore would have a reduced propensity to develop intraocular complications such as the formation of epiretinal membranes. So just to give you a brief overview of what EMT is, so when you have a retinal detachment, your RPE cells, endogenous RPE cells, would be dislodged. Some of these may migrate and proliferate to the surface of the retina as they undergo EMT transformation. And these membrane formation, as I alluded to earlier, sometimes from epiretinal membranes, or in more severe cases, can contract and cause a redetachment in the case of a proliferative vitreoretinopathy. We wanted to see if our transplanted cells have a reduced propensity for this to happen. And we addressed this again by doing immunohistochemistry with comparing pre-transplanted plants and post-transplanted eyes, and also comparing to the RPE in the adjacent site of the transplanted eyes. So here we looked at EMT markers such as COL1A1 as well as alpha SNA1, comparing pre-transplanted to post-transplanted as well as the surrounding NHP RPE cells. Here you can see that in the post-transplanted eyes, there was hardly any increased expression of COL1A1 and alpha SNA1, indicating that these EMT markers are not expressed in vivo. We also wanted to look at gliosis in the overlying retina. So here you can see that in the patients, in the monkeys in which we have transplanted PET scaffold alone, there was increased expression of vimentin as well as GFAP, indicating gliosis. This was not present in the sham retinal blood model, so we know this is not induced by the surgical model itself. And interestingly, in the PET scaffold and adult human RPE transplanted eyes, we see that there was a reduction in the GFAP as well as the vimentin, suggesting that the presence of the cells actually reduces the gliosis in the transplanted eyes. So in summary, we have developed a surgical technique for RPE monolayer transplantation in monkeys, which we can also easily transfer to into humans for clinical trial. We are able to show that these transplanted adult human RPE monolayers can recover in vivo and maintain healthy photoreceptors for up to 3 months. There was no evidence of EMT nor gliosis in the overlying retina, suggesting that these monolayers may be safe and useful source for RPE cell replacement therapy. And Tim, with his team, is looking at running soon in the near future first-in-man clinical trials in New York using this cell resource. The second project of which we're going to focus now on is the cord lining-induced pluripotent stem cells that we are developing in collaboration with Prof. Lim Ka Leong from NTU as well as Cell Research Corp. So just to give you a bit of a background, so these are cord lining cells that are derived from together with Cell Research Corp, and they own the patent for all therapeutics arising from cord lining cells. So we reprogrammed these cells to induce pluripotent stem cells, and we call them CLIPS, so cord lining induced pluripotent stem cells. Some of the advantages is that it's an unlimited resource. It's somatically pristine because you're harvesting them at age 0 versus taking skin fibroblasts from aged patients. So theoretically, there'll be less mutations. There is also a propensity for them to be HLA matched if we really wanted to reduce immune rejection post-transplant. So Prof. Lim's group has essentially shown in a Parkinson's model whereby they have differentiated their clips into dopaminergic neurons and transplanted them into mice models. They've shown that these clips are able to survive better in the mice models in the absence of any immune suppression compared to ASF5 here, which is a skin-derived induced pluripotent stem cells. So in collaboration with them, what we wanted to do is then differentiate these clips into retinal pigment epithelial cells and to assess their immunogenicity profile. So in our experiments, we compared human embryonic stem cells as well as skin fibroblast-derived iPSCs and the cord lining-derived iPSCs, which is labeled as 01 iPSCs. We have developed a directed differentiation protocol to RPE, which I will share with you later. And to assess the immunogenicity, we transplanted them into immunodeficient mice that have a human immune system that has been reconstituted in them, and we call them, these are called humanized mouse models. And then we looked at the systemic response as well as the localized response. So in our lab, we have developed a highly efficient differentiation protocol that allows us to have a yield of at least 95% RPE. It's an 18-day protocol. We, after differentiation, we allow them to mature and accumulate pigmentation. Post-purification, we replate them and put them into transwells or porous inserts. This allows them to polarize. As I mentioned earlier, the polarization RPE is really important for its function because they have a polarized cytokine secretion. And then we also characterize by looking at the mRNA and protein expression, and also look at biological functions such as the ability to form tight junctions, as we can measure using transepithelial resistance, as well as the ability to phagocytose photoreceptor outer segments. As you can see here, by day 24, they have a really nice cobblestone morphology and they have gained nice pigmentation. So what we have shown here is that at the mRNA level, both the ES-derived RPE as well as iPSCs-derived RPE is able to express key RPE mature proteins, such mRNA, such as genes such as PMEL, BEST, and RPE65, and there was the absence of pluripotent genes such as HELP4. At the protein level, we show that they form tight junctions by ZO1. As you can see here, they have outlined a nice hexagonal shape of the RPE cells. For in the pathway of retinoid processing, we see that they express very nicely RPE65 in the cytoplasm. And for ionic transport, we've shown that that sodium potassium ATPase is expressed at the apical portion of the retinal pigment epithelial cells. For functional analysis, we show by FACS, whereby we label photoreceptor outer segments with FITC, and then we feed them to the RPE cells. And by FACS, we show that both the ES as well as iPSC cells are able to internalize the photoreceptor outer segments almost 100%. And we also show that the cells are able to secrete apically neurotrophic factors such as PEDF, as well as basally androgenic factors such as VEGF, as such. And also we look at the ability to form tight barrier functions, tight junctions, as you can see here, measured by transepithelial electrical resistance. They are able to form tight, have good TER values over time, with plateau at about 8 weeks at about 350. So because Prof. Lim's initial evidence suggested that these cells are potentially hypoimmunogenic, what we first looked at is MHC1 and MHC2 expression in vitro, here at the basal level, as well as looking at an induced level by interferon gamma. You can see here that in the cord lining derived iPSCs, there was a reduced basal amount of MHC1 and also a reduced level of MHC1 post induction by interferon gamma. Similarly here with MHC2 post-induction by interferon gamma. To characterize this better, we use the humanized mouse models, as I've introduced briefly earlier. Here we tag our RPE cells with luciferase, inject them into the subcutaneous area of the flank of the mice, and we use both immunodeficient mice as well as humanized mouse. And then we look at the systemic response, measuring serum cytokine analysis and blood T cell profile. We'd also look at the localized response by bioimaging to look at survival of cells by its expression of luciferase, as well as remove the skin plaques to do ex vivo characterization by histology as well as immunofluorescence. And this work was done in collaboration with Qing Feng from IMCD. So first of all, by bioimaging, what we saw was that all three types of cells, ES cells, skin-derived, as well as cord lining cells, are able to survive subcutaneously over time. And when we remove the skin plaques on histology, again, we can very nicely see that the pigmented RPE cells do survive quite nicely in all three cell types. And here in the immunofluorescence, you can see that our violet is RPE65 is stained in violet, indicating that's where the RPE cells are, as well as the staining by OTX in green, indicating where the RPE cells are. And here we can see that in all three cell types, the cells do survive quite well in vivo. We then looked at the systemic immune response, both looking at pro-inflammatory cytokines such as interferon gamma, IL-18, IL-17A, and IL-8. Here, just to orientate you a little bit, blue would be the control samples in which there was no transplant. Red would be the ES cells, green would be the skin-derived RPEs, and purple is our cord lining-derived RPE cells. You can see here, compared to the red and the green, the cord lining-derived cells have a reduced expression of various pro-inflammatory cytokines at the endpoint of 2 months. We then also looked at T cell activation in several peripheral organs such as blood, spleen, and liver. Here, the ES cells are in blue. Red will be your skin-derived RPEs, and the green would be our cord lining-derived iPSCs. Again, you can see that there is a difference between T cell activation in the blood, spleen, and liver in our cord lining-derived iPSCs compared to the skin-derived. So here it seems to suggest that skin-derived iPSCs have a higher propensity to elicit systemic immune response compared to our cord lining-derived iPSCs. Now we're looking at the localized level where we remove the skin plaque and did some immunofluorescence. Here we have looked at human CD45 using a pan-immune cell marker in red, and OTX being the RPE marker to indicate where the RPE cells are. You can see here that in the skin-derived iPSCs, there is a huge infiltration of these red cells, which is CD45, with some in the ES cells and quite little in the CLIPS iPSCs, RPEs. And we quantified this by grading, so 0 being little and 3 being the most. And we've quantified this in several mice. Here you can see that in the cord lining-derived RPE cells that were transplanted, there was hardly any CD45 infiltration compared to a high amount in the skin-derived iPSCs. We also wanted to look at MHC2 expression in vivo because we saw some evidence of a difference in expression in vitro. So here we looked at MHC2 labeled in pink. Here you can see that in the CLIPS RPE, there was an absence of MHC2, but there was a presence in both the ES and the skin-derived iPSCs. And we also looked at CD3 T cell infiltration. And again, you can see that there was a lot more infiltration in the skin and the ES-derived compared to the CLIPS. So much of this actually confirmed the initial response that Prof. Lim noticed in his Parkinson's models. And so a lot of this has been patented. We filed the US provisional patent looking at the use of retinal pigment epithelial cells derived from CLIPS and its hypoimmunogenic properties. And we are also now looking into transplantation of these cells into non-human primates. And we have done a couple of them, and these are some initial results that we have. These are in monkeys with no immunosuppression. So these are xenografts of humans into monkeys with no immunosuppressions. Here you can see that in the CLIP cells, that is the scaffold again with the cells on top. You can see that the retinal structures over it are quite well maintained. There's no obvious evidence of immune rejection. And surprisingly, for a skin-derived, there was a little bit of subretinal fluid, but not much. And this is currently up to a month, and we are following them up to 3 months to look at the structural as well as the functional analysis. And importantly, we are also doing a set of experiments looking at some of the changes of these cells pre and post-transplant. So for these experiments, we transplanted the cells into rabbits' eyes, left them in for a month, and then enucleated them to look at single-cell RNA-seq to look at what's the difference between the in vivo cells versus the in vitro cells. And this is still in progress, and I hope to be able to share this with you in the future. But some preliminary results indicating that the cord lining-derived iPSCs have a downregulation of some of the immunogenic response and upregulation of some of the immunosuppressive response. So now changing track a little bit into retinitis pigmentosa. So RP is a different disease from AMD. In this disease, patients present with a fundus as such, where they've got a pigment deposits in the peripheral part of the retina. And here the primary dysfunction is that actually of photoreceptors. and not really RPEs, is a monogenic disorders, and there are multiple genes that have been identified to be responsible for it. And the prevalence is about 1 in 2,000 individuals, affecting more than 2 million people worldwide. Now, the concept of photoreceptor cell replacement is similar, but not quite compared to retinal pigment epithelial cells, because this field has not been as mature as retinal pigment epithelial cells, because differentiating and maintaining RPE cells in vitro hasn't been as straightforward or simple. So there are 2 clinical trials for these RPC cell replacement at the moment. The first of which, by JCyte, is looking at an intravitreal approach, whereby they inject the retinal pigment epithelial, the RPC cells, the retinal progenitor cells, into the vitreous cavity. And the mechanism by here is that they hope that these cells are able to secrete neurotrophic factors to enhance the endogenous retinal function. The other approach, which is a subretinal injection approach, where we inject the RPCs into the subretinal space, the hope here, of course, is that the RPCs would continue to mature and they would be able to form a monolayer of cells that have the correct orientation. As you can see here, that the photoreceptor outer segments have to be facing RPEs, they have to form synapses with the bipolar layer. And so that's not a trivial task, and expecting these cells to orientate themselves in vivo is not trivial. So it's still not clear, if we were to use a subretinal injection approach, whether these cells can survive, polarize, and also function and have support visual recovery. So the other mechanism in which it might work is that they could also, in a local area, secrete neurotrophic factors to enhance the function of the endogenous photoreceptors. So the current trial by JCyte, it's currently in the phase 2b. They are doing a single intravitreal injection of 6 million RPCs into the eyes of patients with retinitis pigmentosa. In the most recent report, they shared that patients with a central visual field of more than 20 degrees have a better response. And they showed some improvement in terms of best corrected visual acuity as well as a kinetic visual fields. And here, of course, the mechanism is not by physical replacement of the photoreceptors, but through the release of neurotrophic factors such as rod-derived cone viability factors released from rod photoreceptors that would then enhance the function of the endogenous cone photoreceptors. The other approach is by the company called Neuron. They're currently in phase 1-2 trials. The trial started in 2015. They reported that there was some promising efficacy data, but that hasn't been officially released. And so this still remains to be seen if this approach is going to work. And so this comes to our last part of the talk, where we did our collaboration with Rexcell, which is a US biotech company. So Xianmin here, Professor Zheng Xianmin, is the CEO as well as the founder of ACTRIS, and she herself is a neurobiologist. So they published in this paper over here where they used their RPC cells and transplanted into RCS rat and mice and saw that there was a survival of these cells and maturation into photoreceptor precursors. Here, what we did with them was to differentiate their iPSCs, use their cells, the iPSCs that were differentiated into RPCs in the US. They were cryopreserved and flown to Singapore. We further mature them in our lab for an additional 4 weeks, characterize them before we transplanted them into non-human primate models. Here we use both healthy non-human primates as well as diseased non-human primates, where we induce localized disease with cobalt chloride. And then we would do live in vivo follow-up, followed by ex vivo follow-up with immunohistochemistry. So here we did a cobalt chloride disease model. There was a subretinal injection of cobalt chloride that induces HIF-1 alpha activation. This induces hypoxia and tissue damage. So you can see over here, this was the area in which we've injected the cobalt chloride. And on autofluorescence, you can see hypoautofluorescence indicating there was some cell damage over there. And you can see that over time, 1 month, 2 months to 4 months, you can see the loss of the ellipsoid zone. It gets really mottly and no longer smooth, as you can see the multiple layers. If you compare that to the control over here, where you can see a really nice ISOS junction or EZ line, you can see that this is completely gone. So this indicates that there was a significant amount of damage both at the photoreceptor as well as the RPE layer. And here is, we show that after we have induced damage in the eyes, we rescued it with cell injections, and then we followed out for 3 months. So in this area, at 1 month post cobalt chloride injection, you can see that there was a loss of the ellipsoid zone. At 1 month, there was a slight recovery And by 3 months, there was recovery of the ellipsoid zone over there, which indicates there was a certain amount of structural recovery in the transplanted area. We enucleated the eye and we looked for cells that are from the human source by looking at the positivity for markers such as SC121 and anti-mitochondria. And these cells actually co-stain with opsin stainings, indicating that these cells that have been transplanted into non-human primates, these human cells are able to survive and mature into cone photoreceptors. So our future plans within this joint lab is to bring these cells into first-in-man trials in Singapore, because the iPSCs are FDA approved, and now we are working really closely with Retinal as well as ACTRIS to do GMP manufacturing from the iPSCs to RPCs, and then to do the first-in-man clinical trial in Singapore. And also, as I've alluded to earlier, hypoimmunogenic iPSCs for stem cells are really important because they reduce the amount of immune reaction that would be at the local level of the transplant, and would also reduce our reliance on immunosuppressive drugs. And so we are using genetically modified hypoimmunogenic iPSCs within this joint lab, and we hope to help them to develop this and to characterize this in both our in vitro and in vivo models. And of course, as I alluded to earlier, many of these diseases are pretty complex. We're not just looking at the loss of RPE or the loss of photoreceptors. It's actually, we're hoping to replace not just the RPEs, the photoreceptors, but also the Bruch's membrane. So in order to do this, our team is looking into using advanced biomaterials to do co-cultures of RPCs and RPEs, and being able to do that in vitro, in vivo, and hopefully be able to bring this into clinical trials where we can ideally replace not just a single tissue, a single cell, but a segment of the retinal tissue. So I'd like to acknowledge our various funding bodies, particularly for this work would be our NRF-CRP grant that is funding our RPE work, and the IF-ICP grant that is funding our photoreceptor work, as well as the other grants that have been supporting my lab. And I'd like to also acknowledge the various members in my lab who have contributed to this work, and without whom much of this cannot be done. Thank you for your kind attention. I'll be happy to take any questions.
Thank you, Prof. Su. We will now proceed with the Q&A session. So the first question is, may I know what causes the increased number of cells in ONL following the RPE cell transplantation?
Thank you for the question. Yeah, so the increased number, it wasn't an increased number of cells in ONL, it was rather the preservation of the ONL compared to the control, whereby we've only transplanted the scaffold alone without the RPE cells. So in the animals in which there's only the scaffold, but there was absence of RPE cells, the overlying photoreceptors will die off, and therefore the ONL layer will thin off. Whereas in the area in which we have transplanted the AHRPE cells, they are able to support the endogenous ONL and therefore maintain it.
Thank you, Dr. Su. So far, we do not have any other questions. So to our listeners, if anyone else has any other questions, please feel free to post your questions in the Q&A function. Okay, we have another question. So the next question is, what do you think the implications are for the accumulation of toxic materials in an AMD retina for the survival of transplanted cells?
That's an excellent question. Thanks, Bo. I think, that's why a lot of the clinical trials results haven't been overwhelmingly positive or fantastic, because, AMD is a really complicated disease, whereby in the end stage, you've got the loss of RPE cells, the loss of photoreceptors, not to mention all the drusen that is kind of accumulated there and all the toxic materials. So I'm sure there will be implications of the presence of these materials that would affect the outcome of the transplants. So yesterday there was another talk by Prof. Liu Yong in the Stem Cell Society, whereby from China, what they actually do is they remove the CNV first before they transplanted their human embryonic derived RPE cells, with the hope, of course, that you're removing the diseased layer of tissue, and in that process, hopefully remove some of these toxic accumulations and give the transplanted cells a better chance of survival. Surprisingly, they still see quite a significant amount of post-operative inflammation in the form of macular edema. So although probably removal of those materials is helpful, I think there are various other factors. In that case, they are doing a cell suspension. So they are injecting at least 500,000 of cells within the subretinal space, which in itself may induce an immunogenic response also. So in short, there isn't, I think the field is mature, but still there are lots of questions that are still unanswered, and there are various things that we can do to improve the outcome.
Thank you, Dr. Su. We have another question. Do you know how close is your RPC correlated to human fetal retina? Do you think a specific subpopulation of your differentiated cells could lead to better transplantation outcome? Did you perform ERG for the post-transplanted NHP? Does it improve in post-transplantation? Does subretinal injected cobalt chloride significantly disrupt the thin the ONL? Appears that your transplanted cells is integrated in the thick ONL.
Yeah, that's a great question. I think the ultimate measure of the success of transplant is some form of visual recovery. So far, we have done electroretinography. Usually, so far, we have done full field electrography showing that there isn't a widespread loss in retinal function. However, we recognize that that's not ideal. We would ideally need to do multifocal ERG to look at that specific area with induced loss of function post-insult and then recovery of function post-recovery. And this is something that we are working on. We have been able to optimize our settings for multifocal ERG, so hopefully we'll be able to show that, share the results with you soon. And with regards to the earlier question of how are our iPSC-derived RPCs different from fetal RPCs, we are currently in the process of characterizing this because this is also part of the FDA requirements before we bring them into clinical trials. So we are hoping to look at the different subpopulations and the gene expression patterns more closely.
Thank you, Dr. Su. Next question. What are the pros and cons with cell-based gene therapy products for RPE mutations?
Yeah, the question was a bit confusing.
What are the pros and cons with cell-based gene therapy products for RPE mutations?
So I'm not entirely sure what the question is asking, but I suppose you're asking if we were to treat dysfunctional RPEs either with gene or cell therapy, and what are the pros and cons versus gene and cell therapy. I think cell therapy, in my mind, is ideal in a situation where your endogenous cells are no longer there for you to revive. So in the approach of a gene therapy, you would require some cells to be still alive, and therefore you can use gene therapy to rejuvenate these cells. And this is quite evident also in the JSITE study, where in their phase 2B trial, they showed that when they are treating patients with earlier stage of disease, they are able, the neurotrophic factors they are released are able to improve vision better than in patients with lower vision, like a PL vision with hardly any endogenous live cells. So the approach of using gene or neurotrophic factors would have to rely on some cells being present for you to kind of rescue. Whereas cell therapy is, I think, would be more appropriate for conditions, disease in which you have progressed to a later stage where you have hardly any cells for you to rejuvenate. And there the hope is that when you inject the cells back in, either they are able to take over or replace the function of these absence of live cells in the area. Yeah. And also, I guess for cell therapy, it's a gene agnostic approach. So often for gene therapy, you would have to identify the specific mutations, identify the specific patients, and then have a targeted approach. And then the pharmaceutical companies will also then have to develop specific therapeutic approaches for specific kinds of mutations. And then you go into this whole, are they large genes, small genes, are they splice variants, which makes it a lot more complicated. Whereas cell therapy is agnostic of whatever underlying genetic mutation it is. particularly for the neurotrophic approach, where you just inject the cells in and hope that they excrete lots of goodies and they recover whatever cells that are alive there. So there are pros and cons for either approaches.
Thank you, Dr. Su. So if anyone else has any other questions, please type your questions in the Q&A function and Prof. Su will answer your question. Any other questions, anyone? Anyone else has any other questions? We have a few more minutes, 7 more minutes before 9 p.m.
I think there's one question by Tianan, right? In the chat group. He's asking, with the J-site approach with endogenous factors, do we know if the cells persist long-term, and if so, where, at least theoretically? I don't think these cells will persist for a very long time, because when you inject them intravitreally, they will probably die after a while and they will be cleared. Not sure how, either by the trabecular meshwork or by the blood vessels. Yeah, so I think the function of these injected cells are basically like a platform delivery of factors that they secrete to enhance the function of the endogenous cells, and they just die off. So I suspect they will probably need repeated injections in the long term, although the current clinical trial is a once-off injection. Yeah.
If anyone else has any other questions, you can feel free, either type it in the chat box or the Q&A function, that's fine.
Maybe we can release people for lunch.
All right, sure. So if there are no other questions, then let me do my ending off. So thank you so much, Prof. Su. We have come to the end of our lecture. On behalf of ACTRIS, we would like to thank you for your time today, and we hope to see you again in our next, to all our attendees, we hope to see you again in our next lecture on 29th of March. So as you sign off, you will be asked to answer a short survey pertaining to the webinar. We will appreciate any feedback that you have for us, as it helps us to be better at what we do. So once again, thank you very much, Prof. Su, and thank you everyone for attending our webinar. Have a good day today.
Thank you. Thank you, everyone. Thank you. Bye-bye.